Hello everyone and welcome to the biology lecture series, and today we'll be discussing the topic control and coordination. Okay, so this topic is from the class 10th science book. So in the previous lecture we have already studied about the coordination. How can we define coordination? So should be a coordination between the different organ system, then only a particular process can be carried out in the living organisms. Then we studied about the coordination in plants. We studied about some of the terms such as phototropism, photoperiodism, as well as vernalization. All right, then we studied about the difference between phototropism, photoperiodism, okay, and some other differences we also saw. All right, and in the end, in the previous lecture, we studied about the plant growth hormones, also known as the phytohormones. So, phytohormones, they are of 5 types. Okay, first one is the auxin, then we have gibberellins, cytokinin, ethylene, and abscisic acid. So, growth promoters, they are auxin, cytokinin, and gibberellins, and the growth inhibitors, they are ethylene and the abscisic acid. So, in the previous lecture, we studied about the discovery of the growth promoters such as auxin, gibberellin, and the cytokinin. And in this lecture, we'll be studying about the function of these hormones, as well as we'll study about the growth inhibitors. So let's start with the definition of the phytohormones. Okay, so these phytohormones, they are also known as the plant hormone. So they are the chemical substance synthesized by the plants that regulates various physiological processes such as the growth, development, and the response to the environmental stimuli. Okay, we have studied about the stimuli also. There were 2 kinds of stimuli. First one is external and second one is the internal stimuli. Okay, one was external stimuli that arises from outside the body of the living organism, and another one was the internal stimulus. External stimulus can be pain or touch. Touch can be the external stimulus. Okay, external stimulus can be like sunlight and the environmental factors. All right, internal factors, example can be hunger. Okay, hunger is a kind of the internal stimulus. Clear? So this is the definition of the phytohormone. Now let's talk about the type of phytohormones. So there are 2 categories. We can divide these phytohormones into 2 categories. First is the growth promoters and second one is the growth inhibitors. Okay. Now, auxins, gibberellins, and cytokinins, these are the growth promoters, and ethylene and abscisic acid, they are the growth inhibitors. Okay. Hormones, they can be divided into 2 categories. First are growth promoters and the other ones are growth inhibitors. So auxins, gibberellins, and cytokinins, we already know now, they are known as the growth promoters because they promote the growth as well as the development. And ethylene as well as the abscisic acid, they are known as the growth inhibitors. Okay, so discovery of auxins, gibberellins, and cytokinin we already studied. Now we'll study about the functions of auxins, gibberellins, and cytokinins as well as the function of ethylene and abscisic acid. What are the functions of auxins? So auxins, they play a role in the cell enlargement and cell elongation. Now what happens? How they induce the cell enlargement and cell elongation? So these auxins, they induce the cell loosening. Okay, they help to loosen the cells and they increase the membrane permeability also, and they also induce the synthesis of the wall microfibrils. So that's how the cell enlargement as well as the cell elongation takes place. So how will the cell elongate? If the cell becomes a little bit loose and there is synthesis of the cell wall material. What is the cell wall material? Which helps to build the cell wall. So there is a synthesis of the cell wall microfibrils. All right, and the membrane permeability is also increased so that the uptake of material can be done. So all these activities result in the cell enlargement and the cell elongation. Now next role of auxins is in the tissue culture. So in tissue culture, what happens? It is a kind of artificial, you can say, growing of the plants for getting the desired result. So what we do, we select the desired traits, and then using the methods of the tissue culture, we can develop a desired plant that have all the desired characteristics. Now the auxins, they facilitate the cell division, and they also help in the induction of the root, as well as they help in the root differentiation. So this is the role of the auxins. You can say root differentiation, cell division, and you can also say it helps in the induction of the root. Initiation, also say initiation या induction of root formation. Clear? Now, apical dormancy. It also promotes apical dormancy, auxin. Okay, so the presence of the terminal or the apical bud, it involves the failure of the lateral bud growth. So this is known as the apical dormancy. Okay, so what happens? Due to the presence of the apical bud, the lateral growth or the lateral bud formation is inhibited, and this is known as the apical dormancy. Inhibition of the lateral growth is known as apical dormancy, and apical dormancy is due to the secretion of the auxins hormones such as indole acetic acid. Okay, so these apical buds, they synthesize the auxins. And what kind of auxin? Indole acetic acid. So IA is indole acetic acid, and it is a natural auxin. Okay, so auxins can be natural as well as synthetic, but this IA, it is a natural auxin. This is clear. Now, if we want to promote the lateral growth, then what happens? We have to remove this apical buds. So removal of the apical buds, they result in the growth of the lateral buds. Okay, so if the apical bud is removed, then what happens? There will be no production of the auxins. So no auxins are produced, because auxins are produced in the apical buds, you can say, or you can also say apical buds, or you can say the shoot apical meristem, or you can also say the auxins we have seen in the previous class, they are secreted in the apical meristem, either the shoot apical meristem or the root apical meristem. They are also produced in the young leaves as well. Okay, now the removal of the apical bud, so we can say the removal of the shoot apical meristem, it will result in the growth of the lateral buds, because there will be no auxin production. If there is no auxin production, then what will happen? The lateral growth will be promoted. So this is done, okay, in the tea plantation also, where you don't need tall plants. So what they do, they cut the apical buds and the lateral growth is promoted. Now next is the root formation. They also play a very crucial role in the initiation of the root. Now auxin, they can also induce the adventitious root in the stem cuttings. What are adventitious roots? Roots that arise from a region other than the radicle. So how can we define adventitious roots? Roots that arise from the area other than what? Other than radicle. So usually root arise from radicle, but if root arise from some other area, okay, or from the area other than radicle, then these are known as the adventitious root. So what happens in tissue culture also? I have already told you auxin play a role in the tissue culture also. So what happens? It can induce the formation of root in the stem cuttings. So here the roots are arising from the stems. That's why they are known as the adventitious roots. They are not arising from the radical. Now, next function of auxin is the cell division. They also promote the cell division. Now, parthenocarpy. So what is parthenocarpy? Parthenocarpy is the phenomena of development of seedless fruit without pollination and fertilization. Okay, so how can you simply define parthenocarpy? It is the formation of seedless fruit without fertilization. Usually, after pollination and fertilization, the ovary develops into fruit and the ovules, they develop into the seeds. Okay, so this happens after the fertilization. But what happens if you apply auxins, okay, to plants such as grapes, banana, papaya, tomato? So in these plants, you can induce the formation of seedless fruit, and these seedless fruit, they develop without fertilization. So if there is no fertilization, then the seeds which are developed, they are not viable. So in parthenocarpy, what happens? The viable seeds are not produced. That's why the fruits are seedless. Okay, either the seeds are not produced or the viable seeds are not produced. Okay. So I've already told you, after fertilization, the seed will develop. Here, there is no fertilization. Okay, so seeds that are produced are not viable. So this leads to the development of seedless fruit. Now, application of synthetic as well as the natural auxins. So I've already told you auxins are of two types. Natural auxins are indole acetic acid as well as indole butyric acid. Okay, synthetic example can be 2,4-D, 2,4-dichlorophenoliacetic acid, which is also used as a herbicide. Application of synthetic as well as the natural auxin to unpollinated pistils. So pistil represents the female reproductive organ. It represents the gynoecium. And pistil is made up of what? Ovary, style and stigma. Okay, so if you apply auxins to the pistil, then they will produce parthenocarpic fruits, means seedless fruits. Now this phenomenon applies in seed-bearing fruit. Okay, so these grapes, banana, papaya, tomato, okay, these are produced by parthenocarpy. And what will happen? The fruit here will be, which will be produced here, will be seedless. Okay, next is the curvature movement. Net function of auxin is the curvature movement. And this curvature movement we have already seen is due to the differential growth. Okay, so there is differential growth, means there is a difference in the growth. So we have seen if this is the shoot, or you can say if this is the coleoptile, or if this is the stem. Suppose we say this is the coleoptile, and we expose, this region you can say is the shoot apical meristem. Now if this region is exposed to the sunlight, this region is exposed to the sunlight, then what will happen? A curvature will develop here and the tip will move towards the sunlight. In the next stage, what will happen? A curvature will be developed, and this is known as the curvature movement. Okay, and this is due to the differential growth. So what happens here? There is synthesis of the auxin hormone, and what happens? There is more cell division in the one region. So here in this region, what will happen? In this region, due to accumulation of the auxins, here there will be more cell division as well as cell elongation. And this region, there will be less cell division and cell elongation. Clear? So these are known as the curvature movements. The plants, they cannot move, they are still, but the parts of the plant can move. So this phenomena is also known as the phototropism. Okay, so movement of the plant part in response to light, this is known as phototropism. Photo means light, tropism means movement. Okay, now Next role is the abscission. Now, what is abscission first? Let's understand that. Now, it refers to the natural shedding or the separation of the plant organs such as leaves, flowers, fruits from the parent plant. Okay, so you must have seen the mature leaves, the flowers as well as the fruit. They separate from the plant, and this is phenomena is known as the shedding. Okay, so this shedding or the separation, this is known as the abscission. Now, auxins play a role in abscission. So what happens in the young leaves and the fruits? Auxin delay abscission, means auxin delay the shedding of the leaves, flowers and buds, okay, when they are young. Okay, now auxins, but however auxin, they promote the abscission of the mature or the older leaves. Okay, so this is clear. So auxins, when the leaves are young and the fruits are young, then it prevent the abscission or the shedding of the leaves. But when they are mature, then it helps in the shedding of the older leaves by the formation of the abscission layer. So what happens during abscission? There is the formation of a abscission layer. Okay, so if this is a leaf, okay, if this is a leaf, this is the stem, then a abscission layer is formed here. Okay, so this abscission layer is when formed, then what happens? Then it will cut off the nutrients. It will cut off the nutrient supply to the leaves, and because now no nutrients are available, then the leaf will fall. Clear? So this is the role of auxin in the abscission. Now, what happens? It also prevents the fruit fall. So there is a synthetic auxin 2,4-D, which is known as 2,4-dichlorophenolic acid. So this is applied to avoid the pre-harvest fruit drop in oranges and the apples. Okay. So next is the role is in flowering. So auxins is generally inhibit the flowering, but in pineapple it causes flowering. So if we talk about the flowering, so what happens? Auxins, they inhibit flowering, but in some cases such as in pineapple, they will promote the flowering. Okay, now let's talk about the next role of auxins, which is weedicides. Okay, so what are weeds? Weeds are the unwanted plants, okay, that compete with the crop plants. So we have to get rid of them. And these unwanted plants, known as the weeds, they compete for the nutrition, and there is a lack of nutrition for the crop plants. So there is a need to remove them. So what happens? Auxins, they play an important role in this weed control because they act as the weedicide, means they kill weedicides. Okay, or we can also say they act as the herbicide. Herbicide means they kill the herbs or the weeds. So weeds are usually herbs, so they are also known as the herbicides. Now next is the storage. So auxins are also used in the storage. So they are applied for the potato storage. Okay, so this is the role. These are the role of the auxins. Clear? Now let's study about the functions of the gibberellins. Okay, so the physiological effects of gibberellins on the plant growth, they are as follows. First one is the cell elongation. Now, gibberellins causes stem elongation and expansion of the leaves in the intact plants. Okay, this is the major role, stem elongation. One of the major role, stem elongation, as well as the expansion of the leaves. Okay, so you must have seen during the discovery of the gibberellins, we have studied that the rice plant. Okay, so these rice plants, they were infected by the fungus known as the Gibberella fugiculae. All right. So this fungus, it was secreting a chemical substance which was later isolated to be, and it was named as gibberellins. Now you must have seen, okay, or it was observed that these rice plant, they were exceptionally tall. Okay, they were exceptionally tall. Okay, why they were tall? Because of the infection with the fungus Gibberella fugicori, which was secreting a chemical substance known as gibberellins. And gibberellins are involved in the stem elongation as well as expansion of the leaves. That's why these rice plants, they were called as the foolish plant, because these rice plants were exceptionally tall, and this disease, this is known as the foolish, okay, rice seedling disease. Okay. Now, this was the first function of the gibberellins. Next function is the stem elongation. It is also involved in the stem elongation. Okay. Now, these induce stem elongation in genetically dwarf varieties such as pea, maize, as well as the cabbage. And this process is known as the bolting. Okay. Bolting is the elongation of the stem. How can we define bolting? So bolting refers to the rapid vertical growth of plants' flowering stem, often resulting in elongated and the leggy growth. This is known as the bolting, and bolting is because of the presence or the application of the hormone gibberellins. So there is a increase in the, or there is a very rapid increase in the growth of the, or there is a rapid vertical growth of the plant's flowering stem. Why there is the growth? Because of the elongation of the internode. Okay, because of the elongation of the internode. Okay, so stem, they are made up of nodes and internodes. So this is the stem. Okay, these ones are called as the nodes, from where the buds and the leaves will arise, and this area, this is known as the internode. Okay, so there is a rapid elongation of the internode, which causes the rapid elongation of the stem. Okay, the next is the seed germination. So seed germination, so these gibberellins, they induce the seed germination. How they induce the seed germination? By breaking the seed dormancy. Okay, so this is also a very important function of the gibberellins. So they are used to for the induction of the seed germination. And how they induce the seed germination? By producing some chemical messengers. Okay, they produce some chemical messengers. Okay, then which then later on interact with some other molecules. Okay, and it leads to the germination of the seeds. Clear? Now, next is the flowering. What is the function of, or what is the role of gibberellins in flowering? So these gibberellins, also known as GA, they influence the flowering differently depending on the species. So gibberellic acid, they promote flowering in long day as well as the biennial plants. So long day plants, we have already discussed what were long day plants. So these plants, they flower when the day length is more than the critical photo period. These are known as the long day plants. Okay, long day plants, these are those plants that flower when the daylight, you can say, or the day length or the day period is more than the critical photo period. I've already told you in the previous lectures what was long day plant, short day plants, and day neutral plant. Okay, so these are known as long day plant. Clear? Now what happens? Let's again understand a little bit about long day plant. So if this is the critical photo period, then what will happen if the day length is more than the critical photo period? So if this is the day length, okay, day length here is more than the critical photo period, then in these condition it will flower. Okay, and biennial plants also. So what are biennial plants? So they complete the vegetative, so they complete their life cycle in two stages. In first stage there will be vegetative growth, and in second stage the reproductive growth starts. So flowering will start. But these gibberellic acids, they inhibit the flowering in other plants such as the fruit trees. Clear? Now parthenocarpy. Let's study about the role of gibberellins in the parthenocarpy. So we've already studied what is parthenocarpy. Parthenocarpy is the formation of the seedless fruit, okay, which are produced without pollination as well as the fertilization. So auxins also, they promoted parthenocarpy. Now let's study about gibberellins. So gibberellins also, they stimulate the growth and development of the ovary. So we have already discussed ovary develops into the fruit and ovules develop into the seeds. So gibberellins, they also promote parthenocarpy. Okay. So here I've written they stimulate the growth and the development of the ovary into the fruit, even in the absence of pollination as well as the fertilization. So simply we can write gibberellic acid, they induce or they promote parthenocarpy. Okay. So because of this, again, seedless fruit will be produced. Example, I've already shown you tomato and the grapes. They are some of the plants in which seedless fruit can be produced using auxins as well as gibberellins also. Clear? Now, next role is the counteract dormancy. Okay, so we have already we have talked about that it promote the seed germination. Gibberellins, they promote the seed germination. How they promote seed germination? By inhibiting the seed dormancy. Now, the natural dormancy of the buds, tuber, rhizomes, and some seeds, it can be overcome by the gibberellins. And I've already told you, so when gibberellins are applied to the seeds, buds, or the tuber, then what happens? A chemical messenger is produced. Then this chemical messenger interacts with other molecules present in the cell, and finally it leads to the seed germination. Now, next role is the induction of the maleness. So these gibberellic acid, they induce the formation of the male flower on genetically female plants. Example is cucurbits. So a gibberellic acid known as GA3. So GA3 is the most common gibberellic acid that is used, and it induces the male flower. What is the induction of the male flower called? So if the male flowers are induced, then we can say maleness is induced. So initially these plants were genetically female. Okay, means they were producing the female flowers, but you can also artificially induce the maleness using the hormone gibberellic acid. And within gibberellic acid, you can use gibberellic acid 3. So there are a lot of gibberellic acid 1, gibberellic acid 2, and gibberellic acid 3, and so on. Okay, but GA3 is the most commonly used gibberellins. Clear? Now, next role is the induction of the aerial stem. This we have already seen. So what happens? You can say aerial stem means we have, why there will be an induction of the aerial stem? Because there is stem elongation, there is cell elongation as well. Okay, and these all lead to the induction of the aerial stem. So M stems are usually aerial only. All right, so what happens in aquatic plants also, you must have seen, okay, if the stem is below the water, then application of gibberellins will induce the formation of the aerial stem. Aerial stem means above the ground, you can say. Now next is the increase in size of the fruits. So this is also a very important function of the gibberellins. So we can also say gibberellins, they have the commercial importance here. One, how it has a commercial importance? First one was the production of the seedless fruits. Okay. Similarly, auxins also, they were also involved in the production of the seedless fruit. Next is the increase in the size of the fruits. Okay. So what happens? The application of gibberellins, they increase the bunch length, okay, as well as the fruit size in the grapes. How this bunch length as well as the fruit size increase? Why? Because gibberellins, they induce the cell elongation. We have already seen it helps in the elongation of the cell. How? By loosening the cell, as well as by increasing the membrane permeability, as well as third one was by the production of the cell wall microfibrils. So if the cell is elongated, then what will happen? The size of the fruit will increase. And one of the example is grapes. Another example is watermelon. So in watermelon also, you can increase the size, okay, of the watermelon by the application of the hormone such as gibberellins. Clear? So till now we have studied about the role of auxins as well as gibberellins. So these are the two growth promoters, okay, that are used in the tissue culture also, okay, and they are a lot of commercial importance also. Clear? So this was the end of this session, and the next session we will be discussing about the function of the cytokinin, okay, as well as the ethylene and the abscisic acid. So cytokinin is also a growth promoter, whereas ethylene and abscisic acid, they are the growth inhibitors. So we'll study about the roles of all these three hormones in the next session. Okay, hope you enjoyed this session, and thank you very much for listening.